Hello, in this video we will discuss about the transforming growth factor beta. It is also known as the TGF-beta for short. So let's begin to understand first introduction and what is the TGF-beta. So the TGF-beta is highly pleiotropic cytokine. Cytokine means the cell proliferation as well as the cell trigger the cell proliferation and the recruitment of the cell as well as the proliferation basically that play an important role in wound healing. And wound healing means the injury of the skin will be healed. Angiogenesis, it means the blood vessel formation, and the immunoregulation and cancer. So the cancer progression also through a TGF beta. On the other hand, the cell of the immune system produces the TGF beta 1 isoform, which express the powerful anti-inflammatory function and master regulator of immune response. And the embryonic development, cell proliferation, cell differentiation, angiogenesis. So that's all things is due to the transforming growth factor beta. So it is the isoforms can be transforming growth factor beta 1, beta 2, beta 3. That is expressed primarily in mesenchymal cell. So the mesenchymal expressed primarily in the mesenchymal cell. And after this, the mesenchymal cell will proliferate and to produce the different type. So just we will discuss and recap an overview about the transforming growth factor beta, and we will also understand the pathway of the transforming growth factor beta after the binding with the cell receptor. So here is the monocyte, which that is monocyte is contain a TGF-beta receptor, and it is the inactive form of the precursor of the macrophage. And here is the stem cell. And after the binding with the TGF-beta, after the binding the paracrinally or autocrinally bind to proliferate into the macrophage, while the TGF-beta 2 is bind with the stem cell to proliferate and differentiate and maturation to produce the stem cell into the nerve cell, epithelial cell, hepatocyte, osteocytes, and adipocytes. And there are many types. A muscle cell. So in this way, there are a lot of cell differentiation due to the transforming growth factor beta, because it is transform the growing cell into the another form during the cell cycle. So important to know that. Let's begin to understand through epithelial cell. Epithelial cell contain also the stroma in the another part. This is the intracellular and extracellular matrix. This extracellular matrix contain a different microenvironment. These microenvironments contain different type of cell. For example, the T regulatory cell and the tumor associated macrophage, T cell, B cell, T regulatory cell, natural killer cell, and macrophage, and many more. This microenvironment will produce that TGF beta. After the production of the TGF beta will bind with itself, the cell, and after the binding with itself to autocrinally to proliferate and differentiate. And on the other hand, it can be bind with another cell. The neighbor cell is known as the paracrine. So after this binding, it means the transforming growth factor beta is stimulate the cell proliferation, angiogenesis, and immunosuppression inhibition. And in this way, the proliferation of the immune cell will be occur. But important thing you should need to understand that the apoptosis inhibition will be occur. It means the cell will be inhibit the apoptosis. How it's possible? We will also understand about the cellular mechanism and the cell signaling process. So this angiogenesis formation, and here is the proliferation of the immune cell, as well as, and on the other hand, important thing is that the epithelial cell is proliferate and convert into the basically epithelial to mesenchymal transition. Transition mean it can be reversed, and the mesenchymal to epithelial transition is also possible, and this phenotypic transition between states are not binary. So it is not binary. It means it can be in reverse when the transforming growth factor will not bind more. But no, I hope you are getting this concept. But let's begin to understand with the cellular signaling process. So this is the cellular signaling molecule will bind with the ligand receptor. For example, this is the ligand. So let's begin to understand from the plasma membrane and extracellular matrix which that's contain the TGF-beta. On the other hand, the nucleus, and we will also learn about how the gene activation for the cancer and how to suppress. Basically, it is suppress also for the cell division, but when the suppress the cell division, so the proliferation will begin. So this is the nucleus and this is the extracellular matrix. Extra mean the above of the cellular. So the extracellular, it is not the cellular part, and the inside is the intracellular matrix which contains cytoplasm. So here is the receptor. This is the TGF-beta receptor which contain a tetrameric protein. And after the here is the extracellular domain and intracellular domain. Extracellular domain is used for the binding of the ligand. For example, the TGF-beta ligands, TGF-beta 1, activins and nodules. This is the type of the TGF-beta. After the binding, the phosphorylation will be occur, and the cascade of phosphorylation events will be occur. In this way, here is the activation of the SMAD protein 2 and 3, and it will be activated to produce the activation form is the SMAD2 3 complex phosphorylation. After this phosphorylation, this is basically the SMAD4, which that will be bind and to produce the SMAD2 4 3 complex formation. After this complex formation, it will be used for the transcriptional factor we will discuss also. But let's begin to another talk about the another here is the transcriptional factor binding. But let's begin to understand another mechanism, another pathway. So here is the row A. This is the row A protein is used for the activation of different we will discuss also. But here is the TAC1 and MLK3 and mitogen activated kinase 1 protein. This will be activated to activate the further the mitogen kinase 3 6 and mitogen kinase 4, which that will be activated further is the protein 38 and genus kinase will directly activate the SMAD 2 and 3 complex. And after this, here is the on the other hand, the PI3K and AKT and mTOR pathway. mTOR will inhibit the glycogen synthase kinase 3. And here is the SMAD2 and 3, which that express of TGF-beta target gene, for example, RAS and mice. And this target gene basically is RAS and mice, the tumor growth genes, which that will be inhibit. This is the down regulation, but here is the SMAD4 used for the activation of the P15 and P27, which that will be inhibit the cyclin D, and the cyclin D will inhibit, it means inhibition again the retinoblastoma protein, and the cyclin D, CDK cyclin dependent kinase 4 and 6, which that is inhibit the RB, and this RB retinoblastoma protein will inhibit the E2F. And in this way, the all mechanism is used for the cell cycle in progression. So this all mechanism will be inhibit. So the cell cycle in progression will be inhibit. But the glycogen synthase kinase 3 is also inhibit the cyclin E. So that's all things the cell cycle in progression will be inhibit. And the maturation and differentiation and proliferation and specialization will be inhibited by the cell cycle. So the cell cycle will inhibit. So the maturation, differentiation, proliferation and specialization will be triggered by the mTOR pathway. So here is the row A will activate the MDIA and the ROC protein. So this is used for the epithelial to mesenchymal transition, as well as another. On the other hand, here is the fast receptor which that is used for the apoptosis pathway. So it will be inhibited due to the inhibition through mTOR pathway. So that is the mammalian target of rapamycin. Seen will inhibit that fast ligand binding with a fast receptor. So thanks for watching. That is the proliferation through TGF beta. Bye.